So hello again, everyone. My name is Ben Adam. I'm a renal pathologist at the University of Alberta Hospital in Edmonton, Canada. And on behalf of the Renal Pathology Society, it is my honor to welcome you to the fourth session in our latest webinar series focused on focal segmental glomerulosclerosis and APOL-1 nephropathies. So as I've mentioned at our previous sessions in the series, our understanding of these diseases has rapidly expanded in recent years, and these new insights have significant implications for renal pathologists, nephrologists, and patients with kidney disease. And the intent of this initiative is to provide a cutting-edge update on these important topics to our colleagues and partners around the world. And I'd like to acknowledge that the series is supported by an educational grant from Vertex Pharmaceuticals, so thank you very much to them for their support. So a few quick housekeeping items before I introduce our speaker for today. So we have made arrangements for live interpretation again of the session into Spanish and Chinese. And you'll notice the slides have also been annotated for this session with some additional Chinese characters. So we're trying that out for the first time. So just please be sure to select the language that you're interested in hearing within the controls in Zoom. Second, we should have time for a short question and answer session following the presentation. So please enter your questions into the Q&A section and we'll do our best to address as many of those questions as we can. And finally, you'll be asked to complete a brief survey at the end of the webinar. So please take a minute or two to fill this out, as this feedback is very important for planning future sessions. So without further ado, it is my great pleasure to introduce today's speaker, Dr. Laura Mariani, who will be discussing therapeutics and the role of clinical trials in FSGS. So Dr. Mariani completed her medical school training at the University of Michigan and subsequently completed her internal medicine residency, chief residency, nephrology fellowship, and Masters of Science in Clinical Epidemiology at the University of Pennsylvania. She's currently an assistant professor in the Division of Nephrology at the University of Michigan. She has a primary clinical and research interest in observational studies in glomerular disease and is involved in several clinical and translational NIH-funded research studies in kidney diseases, including the Nephrotic Syndrome Study Network, or Neptune, the Cure Glomerulonephropathy, or Cure-GN Consortium, and the Kidney Precision Medicine Program, or KPMP Central Hub. So Dr. Mariani's primary research interest is in developing and applying statistical methods for clinical outcome definition and prediction of kidney disease progression, as well as linking clinical phenotype to novel biomarkers and high-dimensional omics data to better understand disease mechanisms that can be targeted for therapy in glomerular disease. So thank you very much for being with us today, Dr. Mariani, and I will hand it over to you.
Great. Well, thanks so much for the invitation. Some of my favorite collaborations are with pathologists, so I feel like this is a true honor to be here. The other fun thing are the addition of the Chinese characters. I have a middle schooler who decided he wanted to learn Chinese, and he has never, ever before been interested in any of my slides until this round. So thanks for that addition. Okay, so I'll start here with my disclosures. As you mentioned, I'm at the University of Michigan where I work at the data coordinating centers. I do some advising for pharmaceutical companies and then have a variety of other disclosures listed here, including my grant support. So what I'd like to talk to you about today is listed here. So I thought we'd start off with the clinical challenge of FSGS, and I'll highlight a couple patient stories that really made me fall in love with taking care of patients with glomerular disease. We'll next talk a little bit about the standard of care, how we typically treat patients, and what do we do when they don't respond to that initial therapy. I'm going to highlight the astonishingly few number of randomized trials in FSGS. And then we'll spend the rest of the time looking at the future, how we can really improve not only trial design and identification of novel therapies for a condition that's really devastating and we truly have very limited options for at this point. Okay, so let's start out with a clinical story. So I think FSGS is particularly challenging, and these two patients demonstrate that, I think, very easily. So patient A was a 26-year-old man. He was born in the Philippines, otherwise healthy. Early in his 20s, he showed up with classic nephrotic syndrome, really swollen legs. When he came to care, his kidney function was well preserved. And we tried steroids first. He responded beautifully, but would relapse. And so eventually he got on tacrolimus. And anytime he was on tacrolimus, his disease would go completely away. And unfortunately, in the United States, as was common, he lost his health insurance. And if he would stop the drug, the disease would relapse, we'd restart it, and the disease would go right back into remission. Patient B is a 26-year-old woman, African-American. She had been born prematurely, also in her early 20s. She was diagnosed with nephrotic syndrome with proteinuria. She actually presented with a lot of belly pain due to gut edema. But when she was diagnosed, her kidney function was already severely reduced. We did try a few different immunosuppressant medications, but unfortunately, really none of them made any difference in the amount of proteinuria. And actually, by the time I finished taking care of her, she was starting dialysis. On biopsy, these patients both get labeled the same thing. So they both had FSGS based on their biopsy. And they're asking me the same thing. What disease do I have? How did I get it? What's going to happen to me long term? Does this mean anything for my kids? And what's the best, least toxic medication you can give me? And unfortunately, those two patients, although they're labeled the same thing, really don't seem like they have the same disease. And so I don't have good answers really for either one of them for any of these really critical questions. And so I think it's this clinical challenge that really caught me about glomerular disease and feels like we can make a big difference using new techniques in pathology, but other omics technologies to really do a better job taking care of these young, otherwise healthy people. Okay, so pathology, what can I tell these patients? So the conventional classification of minimal change in FSGS, which many of you already know, based on, is based on the Columbia classification, where we look at not only if they have some scarring, but where it is. And the sort of general dogma is then there's this spectrum of how patients will do over time, and maybe minimal change disease patients do better, tip lesion maybe does better, and collapsing does the worst. But in truth, I think I don't have to tell this group here that even within those subtypes, there's a lot of heterogeneity. So the lesion itself can look really, really different. These are all different pictures that Laura Barasoni, who's at Duke, shared with me. But the lesions can look very, very different. There can be variability across the glomeruli, certainly. And then even within those subcategories, this is a sort of landmark paper that looked at longitudinal outcomes in those different categories. What I see when I look at this is I see some tip patients who do badly, and I see a bunch of collapsing patients who actually do okay. And in my practice, I think I see a lot of that what we're kind of taught doesn't always bear out. So I never tell a patient with a tip lesion, don't worry, you'll be fine, because I've had enough tip lesion patients who are very resistant to therapy. So we know this gives us some information, but doesn't give us everything we need. And that heterogeneity translates to not only a problem with therapeutics in this area, but clinical trial design. All right, so let's talk a little bit about what I do and what the guidelines say for current standard of care for FSGS patients. So I think many of you will already know that KDIGO updated their glomerular disease guidelines, and the FSGS chapter is one of the ones that I thought were some really great changes to it that I think better match kind of what most nephrologists do in clinical practice. So because we don't understand the biology of FSGS truly, we're left with using our best clinical judgment to try to, honestly make our best hypothesis about the underlying pathogenesis. And so we know that FSGS probably isn't a disease, that it's really a description of a pattern of injury on biopsy. And there may be many underlying causes of that particular lesion. And so the newest guidelines break FSGS into these sort of categories. So there's what we call primary FSGS. That doesn't mean we know the biology, but we think there's some circulating factor, that these are the patients that often present with classic nephrotic syndrome. They tend to be higher risk, although not all of them recur after transplant. We tend to use other clues on the biopsy like foot process effacement, although It's not always there, that it's diffuse. There are genetic, so this is probably the area we've made the most progress in understanding the biology of FSGS. So there are susceptibility genes like APOL-1, and then there are more monogenic genes that more and more have been discovered. There's, I think, upwards of now 30 genes that have been associated. And with some of the new genetic panels that are available, I think, and the increased genetic testing, I think we're actually learning a lot more about these and about variants within these genes that we didn't have before. So the genetic FSGS, again, I've been surprised often when I find a genetic mutation. So I think our clinical gestalt probably isn't, maybe we don't have a high enough sensitivity for this right now, but certain clues could be obviously family history or other syndromic features that point to a otherwise genetic issue. You know, the response to therapy can be really variable. They're often talked about being more resistant to therapy, and their presentation can be different, can be a little bit less of that fulminant, quick onset nephrotic syndrome. We think for many of these mutations, there's less risk of recurrence. And there's this big group of secondary FSGS. So we know that as kidneys scar for a number of reasons, the pattern can look like FSGS. And so they're listed here, including drugs, infections. Certainly, we've seen COVID recently, adaptive from podocyte loss. And then these patients, they can have true nephrotic syndrome, but often don't. The foot process effacement can be variable, and the risk of recurrence really shouldn't be there if that's the true underlying biology. And then there's a group of patients we just don't know. But when I look across this table, I think what's so hard as a clinician is that aside from the genetic testing, I really don't have a biomarker that proves the underlying biology of why this kidney is scarring and why these people are spilling protein. So I kind of take a good history and do a good exam and hear about what things they've tried and try to kind of make my best guess. But in a lot of ways, it's a little bit trial and error in terms of medication. So what are the, so what I do is exactly this, which is I kind of take all those pieces of history and exam and what you tell me from the biopsy, and I kind of try to weigh them all together. So if someone is really acute onset, lots of protein, very edematous, maybe they have AKI, those are people I tend to think, okay, we should give a shot at immunosuppression. And then there's the people that maybe, you know, they really don't have a lot of edema, it's been slowly progressive, or they have a lot of scar in the kidney that I say, okay, maybe immunosuppression is not for you. And there's many, many things in the middle that I'm not sure we really know how it should tip the scales in terms of immunosuppression. And this is, I think, where the real opportunity is to improve the care for these patients. All right, so what are the current guidelines? So according to the newest guidelines, if you have someone that you've decided you think is potentially in that primary FSGS or immune responsive FSGS, the first line therapy is prednisone. And the guidelines recommend pretty high dose. So 1 milligram per kilogram up to a max of 80 mg per day, or an alternate day therapy. They recommend a minimum of 4 weeks and until you reach complete remission. The maximum is 16 weeks, but I have to say most adults have a lot of trouble doing this. The first month kind of goes okay, but after that, they're having trouble sleeping, they're gaining a lot of weight, and they just don't feel well on it. So it's tough to do 4 weeks of high dose steroids and then a slow taper over 6 months. But I'll highlight that for steroids, the evidence is really not so great. So there are no randomized controlled trials in adults for steroids for FSGS. It's sort of extrapolated from pediatric data. And a lot of the data that supports this is data like this. So this is, you know, I think a good example of a center that published their experience with FSGS. They looked at 25 patients who were treated with prednisone and 26 who weren't, and kind of looked what happened over time. Those who got prednisone seemed to do better, and those who got a longer course seemed to do even better. But obviously that data is not a randomized trial, it's observational data. And so it's subject to all of that confounding by indication. that someone decided they were going to use prednisone and probably kept them on it if it seemed to be helping. And so it becomes a self-fulfilling prophecy. And so we don't really have the data we would want to show that prednisone is helpful. That being said, like my first patient, there are many patients that you put them on a little bit of prednisone and the proteinuria goes from floridly nephrotic all the way down to normal range quickly. There certainly are a subset of patients that do well with prednisone. What's our alternate first-line therapy? It's calcineurin inhibitors. And again, the data for here is also pretty limited. So for first-line therapy, there's again small studies like this one. This was looking at just 6 patients who were started on tacrolimus. But there are a lot of contraindications to high-dose steroids, especially for long duration. So patients with diabetes or psychiatric conditions where you might be worried about that long-term steroid exposure, you can use calcineurin inhibitors, either cyclosporin or tacrolimus. For these, I typically give people about 4 to 6 months. If there's really no response, I tend to stop. The problem is the relapse rate after stopping these is very high. So although the guidelines say you can continue, you could consider stopping after 1 to 2 years with a slow taper. I find if people have done really well, I'm very hesitant to stop it because the risk of relapse is so high. All right, I'm just going to give you a little flavor of our evidence for some of the other therapies that are used. So we already talked about tacrolimus. Again, I showed you that one study that was 6 patients. There's similar studies for cyclosporin from converting from cyclosporin to tacrolimus. Again, small numbers, observational, that show there seems to be an effect of this class of drugs. But again, look down here, you can see really there's a lot of patients that if they make it through steroids and it doesn't work, and then they make it through calcineurin inhibitors, there's still a lot of patients where their disease is not under control. So that's true of tacrolimus. That's also true of MMF or CellCept. There's been a lot of interest in trying this medication as well. And again, small sample sizes looking at it. There was a clinical trial, which I'll circle back to later. But again, you show kind of, there's probably a small set of patients that respond to that, but many of them don't. Similarly, cyclophosphamide, again, this is adapted from pediatric evidence. Again, I'll just highlight the relatively small numbers, 33, 30, 60 here. I'll show you this trial again, where, maybe some improvement in a small set of patients, but the cyclophosphamide group carried some increased risk of infection. So sort of once you get through prednisone and calcineurin inhibitors, the next line agents, including MMF, cyclophosphamide, and rituximab, have some real downsides, and it's really unclear which of the patients will respond to that. So I do end up kind of doing a little bit of trial and error with my patients and just being honest that I don't have good markers ahead of time to assign them to a specific therapy. All right, let's just take a look at a little bit some of those prior trials in FSGS. And I wish I could tell you that there were 10 trials supporting all the evidence and these guidelines, so we really knew that we were helping patients. But actually the number of true randomized trials in FSGS is quite low. And most of the trials in FSGS are actually not an initial presentation. They're at that steroid dependent or steroid resistant disease. And I think that makes sense given that, we know there's a good proportion of patients who will respond to those first line therapies. And it's really once we get out into the disease and they're not well controlled or frequently relapsing, that I think there was a real need to do trials. So I think that's sort of how this happened. So I'll highlight this one first. So this was published back in 1999. This is the group from Toronto, had a multi-center treatment, multi-center trial looking at cyclosporin with prednisone versus low-dose prednisone alone. And you can see in this trial that after 26 weeks, this adult population, those that were put on cyclosporin A good proportion had achieved partial remission, but the black you can see are complete remissions, and it's a really small number had complete remission on cyclosporin. And those who just got low-dose prednisone alone, very small amount had partial remission only, none with complete remission. And those patients, not surprisingly, since their disease continued to be active, those who got the low-dose prednisone alone were more likely to lose their kidney function over time. So the guidelines, that's where tacrolimus and cyclosporin, not only they come in for initial therapy, but they come in for resistant disease, largely based on this trial and some related ones. And like I said, other things that have been tried include those therapies that we've talked about, Cellcept, rituximab, ACTH, potentially combination therapy. I'll highlight the FSGS clinical trial as well. So this was back in published in 2011 in KI. This was children and young adults, 138 of them, again with resistant disease. So they had to have steroid resistant disease, at least a gram of proteinuria, and preserved GFR, I think GFR greater than 40. And they recruited 138 children that were randomized. And at 26 weeks, you can see over here that the dark bars were those participants who were treated with cyclosporin, and the gray bars were Cellcept with dexamethasone. So I like looking at the actual data the way you would as a clinician. And this is proteinuria on top and GFR on the bottom. So you can see there really was no difference between the two arms. And I wanted to highlight this trial in particular because I think the investigators did a really wonderful thing, which was after this trial was completed and it was stopped a little bit early and under recruited. So their goal was to recruit 500 patients. They only got to 138. And they published this really nice article afterwards about what they learned from executing this trial. And I think we probably don't do this enough. We publish studies that do well and we understand, we hit our goals and all those things, but what happens when we don't? And so I really thought if you've never read, it's a really great article. And they talked about the barriers of doing clinical trials in FSGS. And they included things like, the timeline to activate their sites. They talked about contracting and IRBs. They talked about, do we have good estimates at each site about how many patients you actually have that would fit your recruitment criteria. So I think that's really helpful. The last trial I wanted, before I move to kind of a look forward, I pulled this trial to just highlight that problem of heterogeneity. So this was a phase 1 trial of adalimumab in, again, resistant FSGS. It was part of the FONT study group. So really this was a study, early phase, looking at pharmacokinetics and dosing for this TNF alpha inhibitor in FSGS. And there had been good animal model evidence before that there's a subgroup of patients who might respond to TNF alpha blockers. And what I like most about this particular study is this. So these were highly resistant patients. They tried multiple therapies. You can see had a lot of protein to start. So this is their proteinuria values, anything from 4 up to 20, 37 grams of proteinuria. So lots of proteinuria, very resistant disease. And this trial really, you could say, is not successful. Most patients really did not respond to this agent except for these 2 patients. So patient 8 here went from 17 grams of proteinuria to 0.6, and this one here went from 3.6 to 0.6. So I think this issue plagues us in FSGS trials. That if you think back to that chart where I'm making my best guess about underlying biology, I'm not really at true biology. And so when you pool those patients in a heterogeneous way and apply a therapy, you can have a failed trial, but it may not fail for those patients. If we really understood the biology, we might be able to do more successful trials. On that note, thinking about when I look at all of the trials and the data that we have for our therapeutics in FSGS, I think the problem is, number one, what I said, the heterogeneity. We're lumping patients that don't have the same underlying biology, although they have the same pattern of injury on their biopsy. It's rare, so we got to get sites from all over the country and all of the world to participate together. And there's a long disease trajectory. So this is a study from the Toronto registry, and what's on here isn't really as important as looking at, if you just look at renal survival, how many years it takes to see that whether or not your therapy has prevented progression to dialysis. This isn't practical for a clinical trial. And so what we need are good surrogate endpoints. So let's look a little bit towards the future with those barriers in mind. So I'm excited to say that there are actually many novel agents that are under various degrees of study by a variety of different academic institutions, but also industry. So I've just listed a few of them here, mainly because I ran out of space, but there are a variety of different mechanisms. So bardoxolone is a NRF2, sparsentan is an endothelin receptor blocker, as well as an ARB. There's a TNF alpha trial, voclosporin, another calcineurin inhibitor, TRPC5, TRPC6. So as we've learned something new about the biology, mechanisms that may be specific for certain patients, and also mechanisms that may apply across underlying biology, that there are now targeted therapies. And I'm really excited about all of these trials in the pipeline, that maybe instead of just having a menu that only has 2 things with good evidence, maybe we'll start to have a much deeper menu. And I think maybe the medication that's, there's a couple of these that are getting further along, but this one was published back in 2018, the phase 2 study of sparsentan. So this study was looking at both kids and adults age 8 to 75, randomized 109 patients to irbesartan versus sparsentan. And you can see that proteinuria decreased in the patients that got sparsentan. And I wanted to highlight the DUET study because what allowed, I think, this to be a practical study was the development of surrogate outcomes and the use of proteinuria for trial outcomes. So this makes a big difference in terms of practicality of conducting clinical trials in FSGS. We can't wait 20 years to see if our patients end up on to have enough patients end up on dialysis. We need a good surrogate endpoint. And so there's been a recent work in the last really few years looking at how do we use proteinuria? Can we and how should we use proteinuria as a clinical trial outcome? And so this work by John Troost took 466 patients with FSGS who had at least 1 gram of protein. And they had a derivation cohort and then validation cohorts. And what I like about this study is that they looked at essentially not only what should your proteinuria cutoff be, but at what time point could you measure it and have confidence that you're separating those who lost GFR and those who didn't. And they had a mix of observational data, EHR data, trial data. And so what this paper showed was that if you, at 4 months, measured proteinuria, if patients had dropped to less than 1.5 grams and by greater than 40%, you could see some separation in their long-term loss of kidney function. And the same group went back and used that FSGS clinical trial data and showed that probably it may not be an actual hard cutoff as a magic number. There may be a continuous association between decreasing proteinuria and decreased risk of kidney function over time. And certainly I think this fits more with my clinical feeling, which is any improvement of proteinuria, not only patients less likely to have symptoms, but maybe their progression is a little bit slower. All right, so how do we tackle this heterogeneity problem? And so, so I wanted to share with you a little bit of our research that we've been doing in the next study to try to tackle this heterogeneity of FSGS. And one of the best avenues to do that is to start with pathology. So my experience as a clinician is when my patient gets a biopsy and I sit down with my pathologist, they take me on this great tour of the biopsy. And not only do they show me the FSGS lesion, but they show me all these other things. And I always think it's such a miss that that I don't necessarily know what all those other features are that you see, what they mean for my patient. And so the Neptune study has this amazing resource that this is an observational study. Patients are enrolled at the time of a clinically indicated biopsy before we know what they have. Our target diagnoses were minimal change disease, FSGS, and membranous nephropathy. And the Neptune pathologists, led by Laura Barasoni and Cindy Nast, and actually they have a whole big committee, but those are the ones that sort of published this initial approach. We're looking at digital images of these biopsies. So there's a repository of all these. And what the pathologists did is they went through and annotated each glomerulus at each level. And then they scored it. So glomerulus by glomerulus went through all these different features. Do I see foam cells? Do I see mesangial expansion? You guys can probably fill in the list better than I can. But literally went through glomerulus by glomerulus. Does it have this feature or not? They also scored the interstitium. They also scored the electron microscopy. And so what's great about that is that you can then do math with those numbers. So we did this study in Neptune, actually was just recently accepted at AJKD. So we took 221 Neptune patients with minimal change and FSGS by their conventional classification. And then we looked at their glomerular descriptor profiles. So we took that rich, detailed quantitative data that the pathologists had generated. We collapsed all those features down to what percent of their glomeruli have each of those features. And then we can do clustering. So we can just do hierarchical clustering to say, how do patients group together? And I think what was fascinating about this is, so conventionally these are two diagnoses, but we really saw where there were three different groups when we used that rich, descriptive, quantitative data. And so you can see, cluster Z over here, really most of their glomeruli did not have a lot of features. You probably can't project very well, but some of these boxes are gray, showing that some of the glomeruli had these features. There's a kind of a middle group and then a severely affected group. And when we looked at the features that kind of drove cluster membership, they're indicated by these numbers here. So there are things that we, use in our conventional classification, like no or minimal change, but global sclerosis was helpful, deflation was helpful, periglomerular fibrosis was helpful, epithelial cell hypertrophy. So lots of the things that I know you all see and you say to me, but we don't really know what it means. But this was able to highlight that rich description potentially adds additional information. And so you can see that not only did it not perfectly align with minimal change in FSGS, but even that severely effective group, there were some patients that included both minimal change in FSGS in each of those groups. And those patients did differently over time. So those three groups, obviously the group that was severely affected, not surprisingly, they were more likely to lose their GFR over time, less likely to go in remission. But kind of a surprise to me, the other two groups separated as well, that their proteinuria rates were a little bit different. Their loss of GFR was a little bit different as well. And I think the real power of these of Neptune and other initiatives where we're trying to bring together not only clinical data, but biospecimens and longitudinal follow-up is that we can then look at other data domains to try to understand more about the features we're seeing. So for example, in Neptune, we have, at the time of that biopsy, we get an extra core for research, and that core is sent for transcriptomics. And so we have bulk initially Affymetrix data, but then RNA-seq data, where we can look at tubular interstitial and glomerular gene expression data. And so if we go and look for which transcripts are up or down in these different clusters, we can see that not only do those patients look a little bit different on their biopsy and that we can group patients that way, but actually their transcriptomic profiles are unique across these three. And including cluster X, the group that does a lot worse, there's a lot of targetable pathways in here, including things like the endothelin signaling pathway. So having these other data domains, clinical data, pathology, and transcriptional data really allows us to learn where can we pick up information about underlying biology, prognosis, and likelihood of response. So the second example I want to give you from Neptune is, can we not only group patients, but can we, for individual patients, get closer to telling them what's the likelihood they might respond to a given therapy? So in this way, like I said, the power of Neptune is that we have these multiple layers of data. So we have not only patient-reported data, their clinical, their pathology, but we have multiple different types of omic data. So I'm going to show you one that's leveraging, again, that transcriptomic data. So again, this is using the Neptune data, and we took the tubular interstitial compartment gene expression data. So this is RNA-seq data. And what we found is, just using that data, not using any other pathology or clinical information, just asking, if we look at the transcriptomic, how do patients group? And what we see is that in the Neptune data, patients, instead of, again, fitting cleanly in minimal change in FSGS, they actually were in 3 different groups. And we have some other partner networks, ERCB and H3 Africa, that also have similar gene expression data available. And we could show that we actually found those same 3 groups. And when we, again, compare to our conventional classification, you can see that clusters 1, 2, and 3 all include patients that have either minimal change or FSGS. I know what you pathologists are going to say. You're going to say probably some of these minimal change patients were misdiagnosed or missed or undersampled. Definitely possible, but just shows you, like, this was their clinical biopsy, and that's what they were labeled as. So then we could use our other Neptune data set, and we found that one of the clusters, cluster 3, again, did a lot worse over time. So more likely to lose their kidney function over time, less likely to go into remission. And actually, these are unadjusted Kaplan-Meier curves. But we found is, even after adjusting for differences in baseline characteristics like starting GFR and starting urine protein, if we added which transcriptomic cluster the patients belonged in, we actually got additional information. They were independently associated with outcome, even after adjusting for kind of usual clinical parameters. So now we can do a lot more work to try to understand that signature. So first, you could ask, well, where is that signature coming from? So you've got a unique set of genes for that poor outcome predicting cluster 3. And we can go back and ask, well, how are genes different across these 2 clusters? So this is just a heat map looking at genes that are specific for different cell types. So fibroblasts, podocytes, inflammatory cells, and then different nephron segment cells. And it doesn't really matter what to say here, but just to say that the pattern looks really different in cluster 3. And that's actually not just driven by one cell type. So it's not like, oh, these patients have just a lot of inflammatory cells, and that's why their gene expression looks different. So the expression profile looks different across both inflammatory cells and intrinsic renal cells. But now we want to know, okay, great, the expression profiles look different. Are there any specific networks or pathways that are driving that difference? So what we're looking for is, we're trying to use that bulk data that we observe at that single time point to say, can we predict which genes might be driving that difference between the groups? So again, we looked at genes that were differentially regulated between cluster 3 versus 1 and 2. And what we see is essentially genes like this, A and B. And if from the literature, you know that if gene X is activated, A will be up and B will be down, you can infer that if you see this pattern in a group, you can infer that gene X was active. And the reverse, that if you already know this relationship from the literature, you can say, okay, there's A is suppressed, B is up. And so I'm going to make the leap that that gene X, this upstream regulator, is down. So that's the idea. And what we actually saw in cluster 3 was that many of the differentially expressed genes would be tied back to TNF. Not all of them. Certainly, there's many other pathways and networks that were upregulated, but TNF was one of them. And I think that probably also caught our eye because of the prior literature, animal models, the FONT trial, where TNF had been tried in patients with FSGS. So again, we saw data like this, which showed in cluster 3, there's a lot of downstream markers of TNF that are elevated. So, like I said, there are many different genes and networks that are maybe up in 3, but we wanted to know to what extent on an individual patient is their TNF alpha activated. So what we did is develop a TNF alpha score. So from the literature, we have a list of genes that through experimental models, we know if TNF is present, this list of genes will go up or down. And so what we did is create a score based on our tissue transcriptomic data. We took 145 genes, we did a Z-score for each individual gene, and then we averaged the Z-score across those 145 genes. And what that lets us do is for each individual patient in Neptune, we can then calculate a TNF activity score. And these are waterfall plots that show you the Neptune, cohort, and they're color coded by which molecular cluster they were in. So red is that cluster 3. So you can see that most patients that were in cluster 3, their individual TNF alpha score is high. Their Z-score is high. Not everyone. Here's one person over here that it's not. And there's variability within that group, probably because other genes are different. But this pathway actually is not what's separating clusters 1 and 2, but it's explaining a lot of what's different about cluster 3. And again, we had 2 replication cohorts where we could demonstrate the same thing, that patients in that poor outcome cluster were more likely to have a high TNF alpha score. We did some additional work to try to understand a little bit more about not only the score, but potential surrogates for that score. So this is looking at single nuke sequencing from 5 participants that had a high TNF score versus a low TNF score. Again, you can see across cell types, the markers of TNF alpha activation were higher in patients that had a high score than patients that had a low score. So again, giving us some lines of evidence that this is not just inflammatory cells, that there's probably intrinsic renal cells participating as well. And then Jennifer Harder in our lab here at University of Michigan did really amazing organoid work. So, so these are renal organoids, again, exposed to TNF alpha, and we could show if there was bulk sequencing of the organoids, you expose them to TNF, our panel of genes that we were using for that score went up over time, and some of the specific downstream markers, both by transcript for qPCR and measurement of ELISA in the supernatant, so that these downstream markers of TNF, TIMP1 and MCP1 also went up in this organoid model. Because ultimately what we want is to not do a kidney biopsy on every time we want to know what the underlying biology is. We'd love to have non-invasive markers. So for example, we went back to this Neptune participants, we had a urine proteomics data set. And so we could take that individual patient score and try to look for those downstream markers. How well did urine protein concentration, how well was it able to capture intra-renal TNF alpha transcript activation? And so you can, the long-term goal would be that we would take patients, we'd measure their urine for MCP1 and TIMP1 as surrogates for intra-renal TNF activation, and that we could calculate for each individual patient, not their true score, because we wouldn't do a kidney biopsy, but we could calculate a predicted TNF alpha score. So for example, a new patient came in, we have this reference population of a waterfall plot, we calculate their score and we can place them. How likely is it that TNF alpha is a problem for you relative to other patients with FSGS? And to me, this is the dream of clinical trials in FSGS, that what we do right now is we take a heterogeneous group, we put them all in unstratified clinical trials. But what if instead of that, we had a drug where we knew something about the mechanism, and we could profile patients either in their urine or by genetics or some other mechanism that would say, we think this drug is more likely to work in patients with this mechanism. And then instead of doing an unstratified trial, we do a stratified trial. And so this one would fail because only the patients that have that particular mechanism would it be relevant for. Whereas if we stratify, and we know you won't be perfect because we don't really understand the biology, but maybe we increase the probability that the patients enrolled that trial, the drug is actually relevant. And so now a trial that would have otherwise have been unsuccessful becomes successful, and we can see that this drug is helpful. And so I go back to that initial phase 1 trial that otherwise is unsuccessful, except for these 2 patients. But if we had a way to stratify, just increase a little bit the probability that TNF was really a problem for those patients, maybe we change the proportion of patients in this trial that respond. And actually, we're going to test this. So this is an ongoing clinical trial where essentially we're taking patients with FSGS and proteinuria. We are measuring their urinary MCP1 and TIMP1, and we, if they have a probability of TNF above a certain level, then they are randomized here to get anti-TNF therapy, measuring those same biomarkers over time. So this is obviously a hypothesis to test, and so we're excited about this and other trials that are considering patient stratification prior to enrollment. All right, so I will end there. I left my email in case I don't get to all of your questions, but obviously I want to acknowledge the work that I showed is the work of many, many people. Neptune is a great big consortium with lots of various expertise. And so I've listed just a few, not all of the names of the people that have made that possible, but specifically the patients and the study coordinators who take people with, a new diagnosis, scary disease, and they're just incredibly altruistic to give this data so that we can start to learn something more about these disease and develop better therapies. And then lastly, I just want to acknowledge my funding from the NIH that makes my training and research work possible.
Thank you very much, Laura, for that excellent overview and update on the current state of therapeutics and clinical trials in FSGS. To be honest, I had no idea that that the data to support treatment in FSGS was as sparse as it is, but I think that highlights the need for further work in this area. And I think the proposal or the approach that you outlined with improved patient stratification is probably the way to go. So yeah, I'm excited to see how the efforts of your team and others hopefully helps move the field forward. So I just wanted to acknowledge there was a comment about the Chinese character annotations that we added to your slides did make the slides a bit busy, and that's obviously no fault of your own. We did that to your slides. So we'll take that feedback and try to maybe color code it next time to make it a bit easier for people to read. But just wanted to acknowledge that. I'll just start with, we do have a few questions in the Q&A. I would encourage the participants to enter your own questions in, but I'll just acknowledge this comment that that has been entered into the chat first. So just a comment that it would be great if it could be brought back to the histologic features for prediction too. Ultimately, a biopsy is needed for the original diagnosis. So I agree with that. I don't know if you have any additional words.
Yes, I totally agree. So one of the things that we're working on in Neptune is, so I'll just say this. Number one, I get so much information out of a biopsy that I think even in membranous nephropathy, where we have PLA2R, and there's, I think some talk about, do we even need to do a kidney biopsy in patients who are PLA2R positive? I still get a lot of information about how much damage there is, how much scarring. It gives me information about, how good do I expect this person to get? And so you're right. I don't see in any, I know I'm preaching to the choir here, but I don't really see the kidney biopsy is going to go completely away. I think I get so much, especially at disease onset, information that helps me kind of know what direction it's going to go long term. And so even if we pick up other biomarkers that tell us something about biology or stratification that we can use over time, what I would love is that we get everything at the start, and then we can use our surrogates kind of going forward. So that's my comment about sort of where I see that going in the future. And yes, we're doing the same thing. So if we group patients based on transcriptomics or proteomics or other things, the most obvious thing is to say which pathology features can accurately place people in those. So that's useful at the time of biopsy. I think we need both. We need some pathologic predictors of that, and then hopefully some non-invasive biomarkers. Not unlike IgA, where we've got that great calculator that you can apply at the time of biopsy, and then I need one I can use again 5 years later because I'm still taking care of that patient.
Yeah, for sure. Okay, so we do have a few questions in the Q&A. They're mostly treatment related, which is interesting. So I assume these are from nephrologists and not pathologists, but yeah, I'm sure everyone will still be interested in your responses. So the couple questions related to the use of calcineurin inhibitors in FSGS and whether, I guess it should be, whether these drugs should be used in the presence of interstitial fibrosis, specifically in the range of like 20 to 30 to 40% IFTA.
Yeah, it's a great question. Kind of when do we get nervous about causing harm with calcineurin inhibitors versus benefit? So I guess I'll give a couple answers. Number one, that, the good trial from the Toronto group a long time ago looking at calcineurin inhibitors, they didn't base it on fibrosis. I don't think they based the cutoff on GFR. So it was if the GFR was less than 40, they didn't do it. So I think you all recognize that sometimes interstitial fibrosis doesn't perfectly correlate with GFR and vice versa. But I think you're right. That's where I start to get nervous when it's the 30 to 40% IFTA is probably a rule of thumb. I don't always rule it out. And what's good about CNIs is that they're, pretty short acting. So, sometimes I say, okay, why don't we try it? I know that we may get AKI, I may see your kidney function increase, and then we'll just stop it. So sometimes I do, if someone has a lot of scarring, I will try it with that really close. You're going to get labs in a week and a week later, and I'm just going to keep a really close eye on you. I'm going to start really low and titrate up. You know what I mean? I'm just much more careful, I think, in patients that have a lot of scarring. So, but certainly that north of 50%, I think is just not really an option. And then I see the other question about rituximab. Yeah. So rituximab, it is in my algorithm. So I think my own personal algorithm. So if you read the KDIGO guidelines, it sort of says prednisone, calcineurin inhibitors, then try whichever one you want to try next. So it's really not. There's not great guidance about kind of what to do next. So for FSGS, I definitely use it. I think the patients, just like with minimal change disease, the patients that show some improvement with steroids, maybe not complete remission, but partial remission, are more likely to respond to rituximab. There's obviously that really exciting data that's coming out of Harvard from the anti-nephrin. I know that's in minimal change, but I think that's really, potentially really exciting about a biomarker for kind of who might benefit. And there's probably other ways in the future we'll figure it out. So I do try it. I think the caveat is just the last 2 years with COVID. I've been very nervous about using rituximab. I've just had a lot of my own patients on Ritux get sick, so I just, it's just I think a discussion with them about if they want to do that. Luckily, we have monoclonal now. We have more treatments now than we did, years, at the beginning of this. Feels like years ago. It wasn't. Just feels like that. But we have treatments now, so I've been using Ritux a little bit more than I was, say, a year and a half ago when we really just didn't know how dangerous it was.
Then kind of follow-up question, I guess, along those lines is somebody asking for a comment on your approach to management of COVID-associated glomerulopathy, which is kind of related, I guess. But yeah, how much immunosuppression do you use for these patients?
Yes. So I would say it depends. So I have had, I personally have not had a de novo one yet. I've had relapses, and I've actually had a relapse after vaccine. So those reports are out there for sure. So it's a really tough choice for patients because the virus is a risk. The vaccine probably has lower risk, but not zero risk. And so I treated them with prednisone. So I went back and treated them kind of in my same algorithm that we just don't know. Now, is there some small subgroup that if you just treated the COVID with monoclonal, would they get better? I don't know that data well enough. I haven't done that. I just, for my patients that relapsed, I treated it as if they would any other relapse. And, we've seen that a lot, that, patients, there have been cases for a long time reporting with various, viral URIs can trigger a relapse, or other things, and that those tend to respond to steroids. So, so my patients who have relapsed, I gave them a course of prednisone, and then actually, thankfully, they went back in remission. Seen a lot of IgA flares too, I have to say. So, kind of the FSGS minimal change in IgA seem to be the ones that I'm seeing in my own practice.
Okay, thanks. And then there's just this one last question about the use of sparsentan in FSGS.
Yeah.
And you did present the clinical trial data, which is encouraging, but any other comments you can make on that?
So sparsentan, the phase 2 study, I think, was promising to show that it did reduce proteinuria. I think one of the good things about that particular study is that they had learned from some of the mistakes of prior. So that study design, they allowed patients to be on other medications. They had a pretty low urine protein inclusion criteria. So there were a lot of good things about that study design. They also offered open label extension. So if you were randomized to placebo, you could come over to the open label extension. So some things that made it, I think, really good from a trial design standpoint and palatable to patients and clinicians. The phase 3 study called Duplex, they completed enrollment. They are still doing some longitudinal follow-up, and it's not FDA approved yet. I think the preliminary data that I've seen reported at meetings looks promising. I think the thought is that, like many of kind of our newer therapies in nephrology, like SGLT2 inhibitors and sparsentan, some of these other ones, they may be applicable across a range of diseases. And so this problem of heterogeneity may not be as big a problem for sparsentan. I think I'm going to wait for that final study to be published and for the FDA indication before we really know exactly which population this is the right drug for. We need to see like how many patients were using it in combination, how far out were they from their disease, are we trying to get people into complete remission, or is this just a drop in your proteinuria? So I think to be determined sort of how that should fit in our treatment algorithms.
Okay, great. Thanks so much, Laura, for giving this great talk and fielding these questions. I think we'll bring it to a close here. I just want to announce that our fifth and final webinar in this series will be given by Dr. Catherine Lepaitis, also from University of Michigan, on January 27th at 11 a.m. Eastern Time, and she's going to talk about an interesting topic related to FSGS and how we can improve renal pathology communications with these patients and the role of patient and pathologist interactions. So yeah, I wish everyone a safe holiday season, and look forward to seeing you again in the new year. Thanks all. Okay. Thanks again very much, Laura. That was fantastic.
Great. That was fun. Okay, thanks for having me. I really appreciate it.
You're welcome. Thanks again.
All right.
Okay, see you later.
Bye.